want to welcome you to the TAU CDEL General Biochemistry Practical class, of which more will be discussed and conducted in the laboratory. However, within few minutes, I want to run you through some macromolecules that we will be carrying out tests for. The number one on my list are the carbohydrates. By definition, carbohydrates are designated as polyhydroxyaldehydes or polyhydroxyketones. These carbohydrates are contained in foods like cereals, grains, tubers, and fruits. They are generally classified into simple sugars and complex sugars based on their sweetness. The simple sugars are the sweet sugars, while the complex sugars are the non-sweet sugars. Now, let's look at these classifications of carbohydrates one after the other. The simple sugars include monosaccharides and disaccharides. The monosaccharides are the monomeric carbohydrate units, which include glucose, fructose, galactose, glucose, mannose, hallose, and atrose, among others. The disaccharides are also simple sugars formed by the conjugation of two monomeric sugar units joined together by glycosidic linkages. They include sucrose, lactose, and maltose. Now, the complex sugars. The complex sugars include the oligosaccharides and polysaccharides. The oligosaccharides are complex sugars formed by the conjugation of 3 to 9 monomeric sugar units joined together by glycosidic linkages. Examples include dextrine, raffinose, stachyose, fibascose, among others. The polysaccharides are the type of complex sugars that contain more than 9 monomeric sugar units, also joined together by glycosidic linkages. And these polysaccharides include starch, cellulose, and glycogen. However, these are the various texts for carbohydrates that we will be considering when we meet in the laboratory. And here are the basic principles of test for these carbohydrates. Another macromolecule we want to talk about are the proteins. But before talking about proteins, I quickly want us to talk about amino acids. Amino acids are the building blocks of proteins. More than 200 amino acids are identified in nature, but only 20 amino acids that serve as building blocks of proteins are known as common amino acids, or you simply say the primary or standard amino acids. We have a list of these amino acids here. 1. Alanine, arginine, asparagine, aspartate, cysteine, glutamate or glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, and valine. We have their 3-letter codes and their 1-letter codes at the side columns. Now, the tests and basic principles of test for these amino acids are shown in this table. These are some of what we will be carrying out in the laboratory for test of amino acids. Now, proteins, which are another macromolecules, will be carrying out tests on when we get to the laboratory. What are proteins? Proteins are linear polymers of the L-amino acids, or we simply say they are polypeptides. A peptide is formed when two or more amino acid residues are joined together by peptide bonds, as we have in dipeptides, tripeptides, tetrapeptides, pentapeptides, depending on the number of the amino acids that are linked together to form the peptide or the protein. However, peptide bond formation is always accompanied by loss of one molecule of water by a process known as condensation or dehydration. Now we want to look at the classification of these proteins. Proteins are classified based on composition, shape, solubility, and functions. Those that are classified based on composition are simply known as the simple proteins or conjugated proteins or derived proteins. And those that are classified based on shape are the globular proteins or the fibrous proteins. The globular proteins are spherical in nature, while the fibrous proteins are elongated or are in rod-like shape. Based on solubility, this just talks about the fact that some proteins are insoluble in cold water, while some are soluble in hot water. It talks about their solubility in different solvents. Now, some proteins are classified based on their functions. We have some proteins that serve as enzymes. Enzymes are organic catalysts that speed up rates of biochemical reactions. We have some proteins that serve as transport molecules that help in conveying or transporting some other molecules from one part of the cell to the other. There are some other proteins that serve as storage molecules, like hemoglobin, which helps in storing iron 2 or iron 3 ion. Then there are some proteins that have structural function. They help in holding tissues together. For example, the myosin and the actin. These help in structural function. Some proteins have hormone function, such as insulin, which is a peptide hormone. Insulin helps in facilitating the entry of glucose into cells. There are some proteins that are classified based on this function, and some proteins have the function of receptor and immune system. Now, this is a typical test for determining the concentration of protein in any sample. And here we have the procedural steps that we'll be considering, that we'll be using as a guide when we get to the laboratory. Now we want to talk about another macromolecule we will be carrying out a test, or they'll be carrying out tests on. This macromolecule is lipid. Lipids are generally classified into simple, complex, derived, and miscellaneous lipids. The simple lipids are the lipids that contain just glycerol and fatty acids only. And examples include the fat and oil, which are also known as triacylglycerol or triglycerides. Then we have the diacylglycerol, the monoacylglycerol, and waxes as basic examples of these simple lipids. We have another classification, which is complex lipids. These complex lipids contain, in addition to the fatty acids and glycerol, contain phosphate molecule, it could be carbohydrates, and it could be protein molecule. Therefore, we have examples of these complex lipids as phospholipids, which contain, in addition to glycerol and fatty acid, contain phosphate molecule. We have glycolipids, in addition, which contains carbohydrates, sulfolipids, in addition, which contain sulfate molecules, and lipoprotein, which contain both lipid and protein in different proportions. The derived lipids are obtained from either the simple or complex lipids by either enzymatic process, acidic or chemical process. And examples of these derived lipids include fatty acids and other alcohols such as glycerol or sphingosine. Miscellaneous lipids are sometimes not always classified as lipid, but we have some of these hydrocarbon or some of these organic molecules like terpenes that have some attributes of lipids. In short, here is a blood classification that graphically demonstrates what lipids and their classes are all about. Please, we will meet in the laboratory for more tests of this course. Do feel free to drop your questions and comments, which will all be attended to. Thank you very much, but please take note that all these macromolecules that we will be considering in the laboratory have important roles they play in the physiological system.